Again, for everybody else, thank you for joining. I'm Siana Peva, and I'm a junior expert in the Department of Industrial Diagnostics at RIDACOM RTD. And I'm very excited to see you today and share this information about Legionella diagnostics. And I really think it's going to be really helpful for you. A short summary of what we're going to be talking about today, starting with introduction to Legionella diagnostics. What is Legionella? Why it's so dangerous for us? And how to perform diagnostics in water samples based on the 2 ISO standards that are currently for diagnostics of Legionella in water samples that are compliant to the laboratories. Then what is the best current solution that we found for Legionella diagnostics? And we'll also show you a video of the procedure so you can see how simple and easy it is. And we'll talk a little about the differentiation of dead from live cells and why this is so important to perform, especially when doing qPCR diagnostics. We'll also talk about the different procedures for extraction of Legionella DNA, which are compliant to the ISO. Also about water genome units, how are they different from colony-forming units, and how are they more helpful for our research in diagnostics. We'll also see how to generate a report, which is compliant to the ISO 12869:2019. And lastly, we'll talk about how to implement PCR technology, especially if you have no prior experience, and how we can help you in this process. So firstly, a little introduction to Legionella. Legionella are gram-negative bacilli, which can cause very dangerous diseases, Legionnaire's disease and Pontiac fever. Pontiac fever is a flu-like illness, while Legionnaire's disease is a very severe pneumonia, and it is the reason why Legionella is so dangerous for us and why this is such an important health topic. Legionella thrives in water, it lives in water, and they're intracellular parasites of freshwater protozoa. And they also use the same mechanism to proliferate in mammalian cells, especially in our respiratory system and in the macrophages, which are in our alveoli. So the respiratory disease is caused when a susceptible host inhales aerosols which contain the bacteria. And Legionella pneumophila is responsible for 90% of the cases of Legionnaire's disease. However, all of the Legionella species, under appropriate conditions, can cause those 2 very dangerous diseases. Legionella becomes dangerous when it grows in purpose-built systems, such as cooling towers, potable water systems in hospitals, in homes, in hotels, spa pools, etc., where the water is maintained at a temperature of 20 to 50 degrees Celsius. So such water systems, such temperatures and conditions, they encourage the growth of biofilms, which create expanded surfaces for Legionella to multiply. And these biofilms contain other bacteria, algae, or amoeba, and their secretions can protect Legionella. So they allow it to multiply and grow. But here we have to say that Legionella pneumophila has 2 different life states. Non-motile state. The other is the transmissive motile state, which is also very widely known as the viable but non-culturable state, because in this state, the cells cannot grow in culture media, in nutrient media, and cannot form colonies, so we cannot detect them throughout culture methods. So the replicative non-motile state, this is the state where Legionella is in favorable thriving conditions, so it can use all the nutrients to replicate, and it doesn't move. But in the transmissive motile state, this is in unfavorable conditions where the cells are under stress or they don't have enough nutrients. So they transfer into this state, which is because we cannot detect them through the cultural methods because they do not multiply. And here you can see a little scheme of how Legionella is in water, and this is the biofilm where it can thrive, but it also can transfer to the water, and this is the very dangerous state for us because it can be in water everywhere around us. So let's see which are the two ISO standards which specify the methods through which Legionella can be diagnosed. So the first is the ISO 11731-2017, which specifies the culture-based methods. So these methods consist of first sample collection of the water which we need to investigate. Then there's the sample pre-treatment. This includes the steps of acid or heat pre-treatment. And these steps are done in order to minimize the interfering microorganisms, which can prevent us from identifying and seeing Legionella on the Petri dishes and the culture media. So after we pre-treat the samples through these methods, we inoculate them on media, and then we have to wait 7 to 10 days to get the first results. Then the colonies which we think might be Legionella, we have to subculture them and wait for another few days. And lastly, we have to perform confirmation tests to be sure that the colonies are Legionella and certain Legionella species. The measurement unit which is used in this ISO and specified in this ISO is colony forming units, CFU per liter. So this shows us all of the cells that can actually grow, multiply and grow colonies on the media. Then second is the ISO 12869-2019 method, and it specifies the quantitative polymerase chain reaction qPCR methods. So here we have a very different, it's not a microbiological, it's a molecular method for diagnostics. And here the steps are first collection of the samples. Then we also have a form of pre-treatment, but here it is different. It doesn't have to include, and it shouldn't include acid or heat treatment. But we can have concentration of the samples through water filtration. And also this can be done for the ISO 11731. So we can concentrate our samples and bacteria more, and we can detect them easier. So after the pre-treatment concentration of the samples, we have to perform extraction of the DNA from the cells, because if we don't perform this, even if the cells contain the DNA which we want to amplify and detect, the qPCR cannot detect it. So first extraction of the DNA, and then lastly, we have the qPCR procedure. And from here, we get the results in genome units per liter. Here, this is a different measurement unit. The genome units show us one genome unit is equivalent to the DNA which is contained in one Legionella cell. So here, we can detect every Legionella cell in the sample, not only those that can grow colonies and can grow on nutrient media. So here, genome units really help us to be the method to be even more sensitive. So here we can see all the advantages of qPCR over culture method, because there are a lot of differences. So first we have the turnaround time. Here it's a big advantage for qPCR because the whole procedure and receiving the results is in 4 to 6 hours, which allows for very rapid actions to take in order to prevent outbreaks of Legionella. While the culture methods take around 10 to 14 days. So this is a big period of time compared to the qPCR. What about sensitivity? Here, qPCR, it targets only Legionella because we target and can detect and amplify only the DNA of the Legionella. While in culture methods, we can have interfering microorganisms and Also, Legionella grows a little bit slower than other microorganisms, or the colonies can be a little bit smaller, so we can't really differentiate them very well. This can lead to false negative or positive results. Also, qPCR can detect Legionella in every live state. So it can detect it in the viable but non-culturable state. It can detect it in the non-motile replicative state. It can also detect it and detect the cells when they are dead. While the culture methods cannot detect VB and C, viable but non-culturable cells, as said before. And what about false negatives? In qPCR, they are minimized because in culture methods, when we have acid or heat pretreatment, this can be a stress for the cells. So when the Legionella cells are stressed, they can enter the VB and C states. And after this, we cannot detect them because they cannot grow cultures on nutrient media. So for choosing the right qPCR kit and kit for diagnostics of Legionella overall, there are a few things we have to consider, and those usually are challenges when we evaluate different kits. It's a bit hard to find all of the important advantages and factors all in one kit. So first is to consider which pathogens we want to detect and whether we want to differentiate them by species. Because as we said, we have different Legionella species, but the most dangerous one for us is Legionella pneumophila, and especially also serogroup 1. What about inclusion of internal control that evaluates the different stages of the methodology? This is very important for the PCR procedures overall, because we have to check if for example, our sample collection was performed rightfully, if the DNA extraction procedure was also done adequate. And lastly, also if the qPCR procedure and the amplification of the right DNA was also done right. So it's very important to have internal control, and there are different types of controls. Next, it's important to have easy calculation of the results and also to generate reports. If we are following ISO, this is very important, and I'm sure you know that it's very good to calculate the results easy, to have maybe also automatically generate a report with all the things you need. And also for following ISO 12869:2019, here we have to use different standards of Legionella to create the throughout which we can calculate our results from the sample. It is specified in the ISO that we have to use at least 4 standards. And this can be done if we take, for example, a culture, then we can revive it and perform different dilutions. But it would be much easier if you have ready-to-use standards with the appropriate DNA content. And you don't have to do it yourself or have a risk of mistakes. And lastly, it's also very good to just have a convenient, easy-to-use format and undemanding storage conditions. Because sometimes if you have different PCR kits which contain liquid reagents for the PCR mix, they have to be transported in different temperature conditions, and this can lead to difficulties in your methodology. Or for example, if you have to distribute and calculate all the different quantities of the PCR mixes yourself, this can also lead to different mistakes, and it's useful to have them just ready to use. So based on all these factors and challenges, we've evaluated a lot of kits, and we got to the decision that Legionella PCR kit is the best solution, easiest and most also compliant to the ISO for Legionella diagnostics and Legionella qPCR diagnostics. So what do we have here? Firstly, we have detection and quantification of all relevant parameters in one single test. So we have detection and quantification individually of Legionella species, Legionella pneumophila, and Legionella pneumophila serogroup 1. This kit is validated according to ISO 12869:2019, and we also have a validated procedure for DNA extraction. And we can also perform differentiation of dead from live cells. And I'll talk more about that later. It is a very convenient format because we have pre-distributed lyophilized PCR mixed reagents. So the lyophilized format allows for much easier, convenient storage conditions compared to if they were liquid PCR mixed reagents. The kit allows for 96 reactions in convenient 8-tube strip mat. So you can actually use the microplate and break off the 8-tube strips, so you can use them individually. Preferably if your experiment allows for less samples. The results are ready in 4 hours, which is very good compared to if you have to wait a whole 14 days just to have your results. And then we have compatibility with most qPCR thermocyclers because we have different models of this kit with different profile tubes. Next, the kit includes ready-to-use positive control, negative control, and standards. It also contains internal inhibition control, which prevents misinterpretation of negative results due to inhibition of the amplification, and it ensures maximum reliability of the kit. We also have prevention of carryover contamination through the use of uracil DNA and glycosylase. And as mentioned before, we have 4 quantification standards, which are used for generating the standard curve, and you don't have to dilute them or do anything to them to prepare them in ready-to-use format. The 4 standards are A, B, C, and D. You can see here the quantities of genome units per reaction for each one of them. And it's important to know that the first one, quantification standard A, also serves as the positive control, and you will actually see now how this is done. So now I'm about to show you a video of the procedure using Legionella qPCR kit, which involves generating a standard curve using the internal quantification standards in the kit. And then later we'll see the other procedures which can be done with the kit. When you generate the standard curve using these quantification standards, later you can use them for following experiments. And also you can use the kit for colony confirmation, but more about that later. Now we can see the video.
First, you need to program the experimental protocol into the software of the PCR cycler before preparation of the PCR mix. Here, you need to choose quantitative analysis with standards, also choose the correct channels for detection, and enter the number of copies of the controls and the standards. Afterwards, you enter the temperature program according to the instructions of the kit. The second step is to prepare the PCR mix. Take the needed number of strips and place them in a tube rack. Remove and discard the caps from the strips. Pipette 25 microliters of sample into each PCR vessel and resuspend well. If using less than 25 microliters, add PCR-grade water to bring the total volume of 25 microliters. For the positive control, pipette 25 microliters of the quantification standard A, which is the purple cap vial. For the negative control, pipette the PCR-grade water, which is the colorless cap vial. For the standard curve, pipette each of the quantification standards A, B, C, and D in at least 2 copies, again in a volume of 25 microliters. Seal the vessels with the caps. Spin the PCR tubes for 5 seconds at 500 G. Choose the correct placements of the probes in the software, place them in a PCR cycler, and run the program. The results will be ready in 3 hours.
So now we've seen how to perform the procedure for quantity detection using a standard curve with the internal standards used in the kit. Now, what about if you want to use this standard curve for next experiments? We can do that really easily. For example, here we have the generated standard curve, and we can use it for following experiments without running those standard solutions or generating a new curve. We can use the old one. And it's important here that we use the same protocol, but we only run the quantification standard A, which is used as the positive control, as a calibrator. and we run it along with the negative control. So we have a positive and negative control, and we have the generated standard curve from the previous experiments. Now, the next procedure in the Legionella qPCR kit is the procedure for colony confirmation. So this procedure is optimized for rapid confirmation of presumptive Legionella colonies obtained using the microbiological culture methods from the ISO 11731. So the procedure is very similar, and we can use it according to ISO 11731 as one of the different tests that are specified in the ISO after culturing and subculturing of the colonies. Is one of those confirmation tests. So the first step is the same program, the PCR cycler with the appropriate temperature program. And here it's a little bit different than the one shown in the clip before. Then again, the same step, take the needed number of PCR tube strips and check that the PCR mix is at the bottom of the tubes. If it's not, you can centrifuge them. Then discard the caps and pipette the samples and the controls. So we have the positive, negative control, and for the samples, we have to take our colony material from the nutrient media and suspend it. And we have an internal amplification control, which we add all in the sample. Then we seal the tubes with the caps, vortex again, and centrifuge, and we place in the PCR cycler. Again, you have to choose the right placement of the tubes so we get the appropriate results, and then run the program. So these were all the procedures from the Legionella qPCR kit, which you can use for colony confirmation and also detection and quantification using a standard curve. Now, I mentioned differentiation of dead and live cells. So what is this and why do we do it in qPCR? Now, it's important to do it because in qPCR, we can detect every cell of Legionella in every state. So in viable but non-culturable, also in the non-motile state, replicative state, but also when the cells are dead. And when they are dead, and we can and we detect and amplify them and quantify and have the result for the dead cells. Well, the dead cells cannot cause the infection, and detection of their DNA can lead to false positive results. So how do we perform the differentiation of dead cells which cannot cause infection and the live cells, which are the ones we need to have results for because they're the real danger in our water? So here we can simply use the reagent D and the light. It's very simple procedure. We use this reagent, which is a photoactive substance. And then light is the instrument which we use to activate it. It's a type of a lamp with a certain wavelength of the light. So we activate the reagent D, and the way the reagent D works is the following. Here in the DNA extraction procedure, we have our different cells. So we have cells with intact membranes, which are the live cells, and the dead cells, which have their membranes are not intact anymore. So actually reagent D can enter the dead cells, but not the live cells, because it enters through the membrane, in the pores in the membrane. So when it enters the cells, it binds covalently when photoactivated through D light, and it binds to the DNA only of the dead cells because it only entered them. And it actually blocks the amplification of the DNA of the dead cells. So after extraction and during amplification, we have amplification of the DNA of the live cells, but not of the dead cells. So we have the different results here. If we don't use reagent D, we don't perform the differentiation of live from dead cells. We have added result of live and the dead cells, but we cannot know which what part of the result is from the live and from the dead. So we cannot measure the risks here. But with the performed differentiation, we see the appropriate results of live cells, which are the dangerous Legionella cells. So more about the live, which is the instrument which performs the photoactivation. It has a series of advantages. It is fast because it can perform photoactivation of up to 96 samples, which can be processed simultaneously. But it's also versatile because it allows, it has different adapters, so you can use different tubes and plates with it. It's economic with very low energy consumption. It is very convenient because the program starts automatically when you place the adapters and the tubes or the plates in it. It's very safe to work, and it's approved, tested, and certified for CE, FCC, and product safety. So what about the DNA extraction? The DNA extraction for the Legionella qPCR kit is performed through the Legionella DNA extraction kit, which is validated to the ISO 12869/2019. It's a very simple and fast procedure because it uses only one reagent for the extraction, which is the lysis buffer, and it contains a magnetic stir bar for easier homogenization through magnetic stir. So it is applicable to filterable and non-filterable samples, and also for parallel quantification according to the ISO 11731, which is for the microbiological culture methods. So the preparation is performed in a single tube, which allows for minimal amount of handling steps and exposure to hazardous material, and minimizes the risk of cross-contamination. And here it's important to be said that the reagent D treatment for differentiation occurs simultaneously with the extraction of the DNA. So now we'll see how this happens. There are three procedures. Procedure 1 describes the procedure for filtered water samples, while the B procedure is for non-filterable water samples, if they're very turbid or any sample that cannot be filtered. It's not a problem to extract DNA from them with this procedure. And the last procedure enables the preparation of an eluent, which can be used both for PCR quantification and for inoculating and using for detection and quantification of Legionella according to ISO 11731. So for all three procedures, for the filtrated samples, non-filtrated samples, and also for using parallel to ISO 11731, we have the same extraction process. We just have different sample preparation. So now, first, for the pretreatment of filtered samples, we first see the pretreatments of the probes for the filtered samples, and then we'll see the same extraction procedure for all three procedures, A, B, and C. Now, first we have to add rinse buffer to a 5 milliliter tube. Then we perform filtration of our samples, and here the filters have to be 47 millimeters with a pore size of 0.2 micrometers, especially for, exactly for Legionella. So we filtrate the samples, then we add the filter with the top sides on the inside with vortex and centrifuge, and then we're ready to proceed with the DNA extraction, because in the rinse buffer we already have the cells. So next is the procedure C, which is where we can use the eluent for inoculating also nutrient media and for qPCR procedure. Here we again filter the samples, transfer the filter to tube, but here we add a diluted Ringer solution, which is one of the diluents which are specified and compliant to the ISO 11731 for the cultural methods. And in this ISO, there are also other diluents specified, but here we can use Ringer solution or PBS. It's not recommended to use Pages saline or sterile type water because this can negatively affect the qPCR results. Then after this, we again vortex and centrifuge the tube with filter, and we are ready to proceed with DNA extraction, which is for use of qPCR, or we can already use this solution to inoculate the nutrient media or the ISO 11731. And here is the same exact extraction procedure for the three procedures A, B, and C for filtered and non-filtered samples. So first we add the sample, then we perform the differentiation of live and dead cells, which is used performed by adding the reagent D and then treatment with the D light, first in the dark and then in blue light. Then we centrifuge, remove the supernatant, and then we have the step of extraction of the DNA. Here we use the lysis buffer, we prepare it to be homogeneous, then we add it, and we have to mechanically disrupt the cells. We incubate in a heating unit at 100 degrees Celsius for 5 minutes. We mix, centrifuge, and we have the supernatant for detection. So here, within the supernatant, we have the extracted DNA. Now, after we've performed the extraction, we have performed the differentiation of live and dead cells. We've had our qPCR procedure. Now we have the results, but what do we do with them? How do we calculate according to the ISO in genome units per liter? And also how do we generate a report? So the genome unit, as we said, is an amount of DNA equivalent to a single bacterial cell. And we saw that the quantification standards in the Legionella qPCR kit are measured in GU per reaction, and also by the use of the calibration curve, the standard curve. This results in such a value for every sample analyzed, and we'll see how to convert this to the measurement unit specified in the ISO, which is GU per liter. Here we have to know that the limit of detection of the kit is 3 genomic units per PCR reaction, and the limit of quantification is 10 genomic units per PCR reaction. And to calculate the results, you need the CQ values for every sample, for every detection fluorescent channel. And these CQ values are automatically displayed in your PCR software. So you just have to take them and transfer them to this table. So calculating of the results from the qPCR kit and generating a report is performed very easily because you have an Excel template provided with the kit in which you enter the results that you got for your standards in order to generate standard curve and the values, the results you had for your samples. So in here, first in the orange fields, we enter the values for every channel, for the FAM fluorescent channel, the HEX fluorescent channel, and the ROX fluorescent channel. We enter the values for the standards. And here we have to perform them in at least 2 copies. So we enter the values for the first copies here and for the second copies here, and so on for each channel. And here below, you can actually see if the efficiency, the slope, and the R squared are compliant to the ISO. So if they are, it says here passed. So it is compliant for every channel. Now, what about the positive and negative control? The values we enter here. And here we can actually have one mandatory copy and the second is optional. We can also see here if the results are compliant with the ISO. And then in the green fields, we enter the values for our samples. And here you don't need to have them in copies. You can have just one sample. So here you can enter the sample volume, which has to be 25. The DNA dilution factor, DNA sample volume. If you have used RINS before, also specify the volume here. Which procedure for DNA extraction you have used is also specified here. And here, in these 4 fields, you enter the values for each fluorescent channel, the CQ value for each fluorescent channel for every sample. Then here you can see the PCR result is valid. And if you have adequate internal control result. And this is actually all you have to enter in order to generate all your results and report compliant to the ISO. So this is the final results field and generated report. In this graph, you actually can see the standard curve that you generated. In this field, you can add any notes you want. Here, you can specify the volume for which the genome units should be reported in milliliters. Here, you have the names of your samples, sample volume, and here are some measurements that are very important for the ISO. And here you can see the quantified results, and actually you can see here if it is detected or not, and the values. And this is fully compliant to the ISO. So this was about the DNA extraction kit, the qPCR kit, the differentiation of live and dead cells, and the generation of results and reports. But I know, and I saw that in the registration form, a lot of you don't actually have experience with qPCR yet. So if you decide to use this technology, there's a few things you can consider for advantages. And also if you already use, for example, the microbiological methods for diagnostics of Legionella, you can use PCR not only for Legionella, but for a lot of other diagnostic procedures. The remarkable versatility of PCR enables diagnostic laboratories to use just a single device to perform a vast array of assay types. So besides for Legionella, PCR is a very useful technique for diagnostics of Salmonella, Listeria, and E. coli for their detection and identification performed rapidly. Also, PCR is very essential for the detection of viruses, and it's really because there's not a lot of other diagnostic techniques for them. So, example, for norovirus or hepatitis A. And also food safety is another field which qPCR is very important. It plays a key role in detection of pathogens, of allergens, and of GMO. And another important factor and benefit is that you improve your efficiency because, as you saw, the PCR procedures are performed very rapidly. You get very fast results. So you can perform a lot of work more efficient, more fast. And so you can expand your service and perform it even faster. And if you have no prior experience with PCR, we know that it can be a very daunting task to start from zero and have to design the lab, choose the equipment, choose the kits, choose the consumables, have to train yourself if you've never used this equipment and techniques before. But here we really want to help you because we know it can be very hard, but we can use our knowledge and experience to help you in this task. So we can help in every step of the way, starting with the laboratory design, then selection of suitable PCR equipment according to the services you want to provide, diagnostic procedures you want to perform, then an appropriate selection of kits, of reagents, of the right consumables. Also, when you have chosen all of this, we install all the equipment, we perform training so you can know how to use it properly, how to clean the equipment, how to use it for different methods, how to troubleshoot if there are any minor problems, or if there are bigger problems, you can always call us, ask for help. We provide ongoing technical support, so you have nothing to worry about. And also we perform customized project assistance, if there are different projects that you need consultancy for, we can also help. So with this, we've reached to the end of the webinar. So I really want to thank you for attending. It was a pleasure and an honor for me to do this and very exciting. So now if you have any questions, you can ask them now in the Q&A tab or in the chat if you want. And I'll try to ask them, but we don't have a lot of time left. So if there are any questions left, we'll answer them in an individual mails to you. And also, if you have any questions after the webinar, you can always call us, email us, or reach us through our social medias also, where you can follow us to learn more about our events. And I also, again, want to invite you, as I said in the beginning, we'll have workshops on the same topic for the same kits, for working with PCR, if you haven't worked with PCR before, or if you just want to try the kits that are mentioned now, see if they will work for you. We can always talk about your goals for the research that you want to develop, or the diagnostic service you want to provide. So yes, thank you.